you want to download or find information about a macromolecular structure, so say the structure of a protein or of an RNA, or in this case, the structure of a protein bound to an RNA, the place that you go is the protein data bank. It hosts structures that were experimentally determined, as indicated by this kind of Erlenmeyer-Flass symbol, and now it actually has structures that were computationally determined, such as with AlphaFold. There's a lot of things you can find in the entry, including information about the experimental determination. It will also allow you to download the data view and download the data files so that you could use them in, say, PyMol or Chimera X, maybe explore them in more detail than you can with the integrated structure viewer. You get information about the sequence and kind of annotations that have been put onto the sequence. You can even explore these annotations in 3D to see where things correspond. You can also get information about sort of the quality of the structure, both in these slider bars as well as you can actually download. There is this validation report where you can get information, including residue level details, about how well the model matches the data and how good the data is for that model. Much more in extracrystallography and what I mean by model and data and things like that elsewhere. Today I just want to walk you through the basic parts of a PDB entry and how to get what you might need. A really quick overview of what you find on these pages, and then I'll go ahead and step back and go in a little bit more detail about the different sections and where to find what. So on the main page, a structure summary, you get a quick snapshot. You get information about the structure. You get information about any sort of related publications. You get in. So a really quick overview about these different tabs. So a really quick overview of what you'll find in one of these experimental entries. And then I'll go into more detail into the various sections. But first, just kind of a really quick look, and then we'll dive in. So on this main page, you're going to see a quick little snapshot. You're going to see information about the sort of where does this come from, what organism does it come from, where was the protein made, in what organism was the protein made. So in this case, the protein was produced, it was expressed in these insect cells, even though the protein gene was coming from humans. You get information about how it was experimentally determined and the quality of the experimental determination. And so both in terms of the underlying data as well as the model that was built into that data. You get information about any associated publications, and you get information about what is inside of that structural model. So in this case, we see that we have a protein, we have a nucleic acid, and we have a small molecule. Each of these are given different chain numbers, and we'll go into that later. A little bit more information about the experimental data and validation, as well as revision history. In the structure tab, I can go in and 3D explore the structure. I can look at annotations, find information about domains, get links to, find other databases to learn even more. And speaking of learning even more, I get even more of the experimental data information. I can look in the sequence level and look at the information overlaid from various different annotation sources, and also information about the quality of the model in various places. Keep an eye out for gray regions, which tell you that the region is unmodeled. So even though the macromolecule was, the sequence was actually there, the protein was there, the RNA was there, whatever it was actually there, it wasn't, the structure, the data for it wasn't strong enough that you could actually build it in the model. So it was physically present, but unmodeled. You can select which chains you want to look at and explore them here. You can find information about where in the genome the thing comes from. If you had multiple entities, so say you had multiple proteins, or you had a protein that had multiple subunits that were different, so a heteromultimer, you could find information about each of them by coming down here. You can download the files to go explore in a different thing, like a more powerful structural viewer like PyMol or Chimera X. You can download the files here, download them in this PDBX MMZIF format, and use these extended PDB IDs these 12 character long ones. We're phasing out the 4 character long ones and transitioning them to this longer one. More on that in another video, as well as a little bit more later in this video. So now let's step back and dive more in. So this is the structure of Argonaut-2, AGO2, bound to microRNA 20. And I'm not going to go too much into it here. I have lots more elsewhere. But basically, AGO2 is this really cool protein that is involved in RNA interference, RNAi. So it binds to a small RNA. This can be an siRNA or it can be a microRNA. And it uses that as a guide to go find messenger RNAs that encode for different proteins, and it finds the messenger RNAs that have a complementary sequence to that small RNA guide. It can then shut down the production of protein from those messenger RNAs, and so it's a really important form of gene regulation, as well as a growing tool to be able to target specific messenger RNAs for destruction. And so it's this cool structure, and it was actually solved. Well, how can we find out? Well, I know from insider knowledge, but if you go to structure summary, it was solved, or what basically the structure was determined by Lotto Kayam, who was a postdoc and then a more senior scientist in the lab of Lemur Joshua Tor when I was a grad student there, and he was my mentor. And this structure I used a lot in my research. And so you can see here that it was experimentally determined by X-ray diffraction. It's this part of the screen is telling us about sort of the quality of the data compared to other data, so compared to other X-ray structures, as well as compared to structures of similar resolution. So the resolution, much more on it elsewhere, but you want that number to be low. That basically tells you that the electron density map that was this model was kind of like modeled into, that the electron density was fairly crisp. And so if you go over here, you can actually put on the electron density and you can show different things. And if you go, you can actually set it. And so you can see that the density in some regions is crisper than others, as well as how well the model, so basically these sticks and things, how well does that actually fit into that map. So again, much more on that elsewhere, but the information about like how well it fits in things and kind of the geometry of the model can be found but through this like PDB validation compared to all structures, compared to structures of similar resolution. And then if you want that full validation report, you can get that. You can also explore sort of the quality of the model in 3D. You can get to that 3D version when you're over here and you explore in 3D here. Or what you could do is you could actually press this 3D report when you're over here. So that's how you find information about the sort of quality of the model. If you want information about the publication, so there's not always a publication associated with it, and sometimes it'll be like a pending publication, so it'll be like to be published. And sometimes people just put up structures in here that don't actually end up getting published, but then people can still use it. So it's going to give us the DOI, so we can go and find that paper. It gives us the abstract from that paper, various things like that. Note too, it has this like 4F3T. So this 4F3T is this PDB ID. And so this here is the extended PDB ID. So typically PDB IDs, so these like accession codes that you could go and find a structure. So when you're searching for a structure, you could either search from the accession ID if you know it, or you can search based on like the protein you're interested in, maybe the organism. You can do advanced search to search for some really fancy things. But if you're like reading a paper and it tells you the PDB ID, like at the end of the paper often, if it was a paper that solved the structure, it'll have the PDB ID. And then you could go and search for that. So typically these PDB IDs have been four characters long, and they're now expanding them to 12 because they're running out of the 4-character ones. And so you can basically transform one of the older ones into a newer one by putting PDB underscore and then 4 zeros in front of that older 4-character PDB ID. And this gives you the extended PDB ID, which you should start using now. They're going to only be issuing these longer ones in the future, probably around 2029. These longer codes are basically, they're more compatible with the newer data formats, whereas the older ones aren't. And so in terms of the files, if you come over here, you can see the FASTA sequence. That's just going to tell you the sequence of the protein, the sequence of the nucleic acid or whatever else is in here. And so in this case, we have an RNA and we have a protein, and we can see their sequences with the FASTA. If we want the actual structural information, we're going to want to go to this MMSIF this PDBX MMSIF format, which is the new standard, you don't want to use this legacy PDB format. It doesn't have as much information, as well as it is not compatible with the longer codes, and it's not even compatible with some of the larger structures. So just like the PDB, this is a PDB.pdb file. This is the legacy PDB file, and you can see sort of the format like this. It's very set. Whereas with the MMSIF, there's more room for expansion, and you can have longer entries and things. So more on that elsewhere, but that's the gist. And you can go ahead and download those files here. You can also download the validation reports. You can display the files if you just want to see them in your browser with the display. Okay, so that's the PDB ID, the accession code. And you can, again, search for that, and you can typically find these at the end of papers. If I wanted to download the files, that's how I do it. If I wanted to look at the structure in 3D, well, I do that over here. Let's talk about some of the things in the structure. So if I scroll down, I see this macromolecules, and there will be a chain basically for each thing that you have in there. So in this case, this is a protein that has a single chain. And so I only have a chain A. Say I had a dimer, I would have two chains. So I would see an A and then a B. In this case, the nucleic acids, so the nucleic acids are over here, and so they're in the second one. And you can see that when I click on these things, I'm seeing information about them, including the length, where they came from, and information. If you scroll down here too, what you can see is that you have this information about sort of various features of the structure. And so some of this, like disorder regions, hydropathy, so based on whether the amino acids are polar or non-polar. And then one of the most important things that you're going to see is this sort of gray stuff. This is unmodeled. If we go to one of these unmodeled regions, we can see that we can't even click on it because we can't see density there. We can basically see on either side. So when you see one of these dotted lines, that's an unmodeled region. The data in that region was not strong enough to be able to model. There wasn't actually enough electron density there that you could build it into. So when you see those dotted lines, that's telling you those regions are unmodeled, but the sequence itself, like the actual physical RNA, the physical protein, was there in those structure, but the data is not strong enough for you to model it in. And so if I were to go and I were to look at the structure, we can actually go. It by default uses this Molstar plugin. And what you can do is if you actually go and you come over here, you click on some region you're interested in, it'll give you more information, as well as then if you go to density and you go to enable, it is going to give you the option to actually see the density in that region. And if you click on something, it'll take you to that region. You can see there's basically nothing to see here. And so they couldn't build it in, even though they know that that place is present in their sequence. And so if you see those the grayed out residues in here, or you see when you're looking at the sequence, you see these gray things here, that's telling you that the protein was there, the RNA was there, whatever it was actually physically there, but you couldn't see it because basically the signal for it wasn't strong enough in that region. And this is often because you have something that's very flexible in that part, and so you're not able to actually see it. So often it's really important that you go and take a look and see what is unmodeled, and make sure that's not a region that you're really interested in. If you were trying to then say, use the structure to model a drug binding site if the site isn't actually visible in the structure. For things like that, you might need to use predicted structures, but use them with caution. And so basically the darker, there's a lot of regions, say, in this protein that are not very, very soundly predicted. And again, this isn't just like it's the computer's fault. It's just that proteins are really, really floppy and flexible. And sometimes there isn't really a distinct structure, or sometimes the distinct structure doesn't come into place until it actually binds to something. Even if the structure is modeled in, that doesn't mean that it's modeled in well, and it doesn't mean that it's trustworthy if you want to go and, say, model a drug into it. And so the way that you can find more information about kind of like how trustworthy the data is in a region is in the PDB structure validation report. So remember, you could look at that in sort of 3D form and see which regions are not very, might have some issues, or the data wasn't very strong in those regions, as well as then go and look at the validation report and look at the residue level. It kind of like it codes things in terms of how well the model fits the data. So this RSRZ It'll give you things about geometric problems. So maybe the angle is a little weird for a normal amino acid. Maybe there's clashes, so these amino acids are trying to occupy the same space. That's the sort of thing that you want to look into if you are really worried about a specific region that you're interested in. Make sure that it's actually strong. So that's again one of those kind of like more technical things, but something to know where it is. You also want to look and see like what's the B factor like, and so that's kind of like the thermal movement. So how kind of floppy is a region? And so actually if you go over here and you go to this polymer, and if I go and I want to set coloring, I can color it by the atom property, and I can color it by this like uncertainty disorder, and it'll tell me a little bit about that, and about sort of red is regions that maybe are more flexible, more floppy. The amino acid, the data in that region was suggesting that those regions are more flexible. And again, much more on these sorts of things in other places, but that's just something to keep in note and something to know that's there. But when you're in an entry, basically if you go over to the sequence tab, this is where you can find a lot of really cool information, including about physical properties of the various amino acids, as well as things about the structure itself. So like where there are unmodeled. There's a lot of technical stuff I'm not going to go into, but you can find a lot of that information here. And you can see things about secondary structure, all sorts of really cool stuff. And it'll also take you to the UniProt entry. So lots of cool stuff there. And again, you could also do that in the 3D one we were looking at. You can get information about where in the genome the protein information is. So you can see that this is basically on chromosome 8. And this is where you can find it. And you could even, when you were looking at the sequence, see information about various splicing. So again, there's lots of information. But often all that you're going to need to do is go to the structure summary, get this basic idea, look and see what there are in terms of what's inside of here. So we saw there was the protein, we saw there was the nucleic acid, but maybe that's not all. And in fact, if we go down, we can see small molecules. And so in this case, there was a molecule of phenol that was bound in there. And it's a pretty cool story is that the phenol actually binds in the place where a tryptophan binding pocket in the protein that binds to tryptophan residues in GW182, which is one of the proteins that kind of helps recruit the things that shut down the translation from that messenger RNA. So cool biological stuff, but yeah. Okay, if you want more information about the structure, so there's a whole structure experiment tab, and there's also this experimental data and validation where you get the gist. So in this case, because it's an X-ray structure, you get this information about the unit cell, about the space group, about the software used. And then you can also see information about the entry. So sometimes they have multiple releases because you can kind of re-refine the data. And so, yeah, and that'll be found there. And then if you wanted more information about the experiment, including the conditions for the crystallization in this case, you could see it here. And you can see that phenol was included, and that phenol, then you could see it in the structure. Again, you're going to get a lot of information about the various atom content and things. Note too that if you're on one of the computationally determined pages, you're not going to see quite as much information. And so you're not going to see that experimental tab. You'll still get a structure, but you're not going to get that option to see maps. What you'll see instead is you can see things like how confident the structure is. Annotations, it'll still annotate it with information from various databases. So in this case, it tells you about that it's kind of like been found to bind to chemokines and things. If I go to this one, well here when I go to annotations, you see there's a lot of things about various domains. And you can also go to find various folds. And there are actually ways in the PDB where you go to advanced search, where you can actually go and you can search for things with a similar folds or with a specific fold. And you can find a lot of links to various databases that are going to tell you even more things. And so again, if you wanted to do an even more advanced search, you could come over here. Okay, so back to the structure. Let's just review the main things. So we come to this first page. If you get lost, come back to this first page. This is where you're going to be able to download the structure files that you want. So remember, you're supposed to be now switching to this PDBX MMSIF format, and that is going to use these longer PDB IDs. If you have an older ID, just add PDB underscore 4 zeros and then the forward character one, and then you get the longer one. Note too that it's helpful to kind of write the IDs in the lowercase for the text, because then you can tell apart things like I's and L's. And so try to get in the habit of doing that. In this case, this is also in this nucleic acid knowledge base, which you can find here. The PDB also has information from like the EMD, so this has information about electron microscopy structures. The worldwide PDB, they have a lot of really great information, and so this is the RSCB PDB, so it's the Rutgers one, the US version. There's also versions in Japan and Europe, and so you can find basically lots more about the PDB here. Okay, and yes, so then you scroll down, you see on the left, you see information, you kind of get a snapshot of the structure. If you scroll down, you get information about the publication associated with it, when applicable. You see information about what is in there. So you'll see proteins, you'll see nucleic acids. You can see that they're given different chains. And so in this case, we have a protein as chain A and a nucleic acid as chain B. But sometimes you might have multiple protein chains, and so you could have like A, B, C, D as protein chains. These chains is going to be how you can kind of refer to things when you're going and you're looking at the structure, either here, when you want to basically do it here, and you can select different things that you want, or when you're in a more powerful molecular viewing software like PyMol or Chimera X. So again, this is the validation information, kind of telling you about how well the data, how strong the data is in terms of the underlying electron density maps, as well as how well the model fits the maps, as well as how well the model fits geometric, like normal geometry things, and clashes and all that good stuff, which I go into much more elsewhere. It'll also tell you about small molecules that are present, and it'll tell you more about the data. And so the experiment, and you want to find even more about the experiment in the experiment tab. You can find more about the protein, about the domains, all that stuff in the annotations tab. You can find sequence level information in the sequence tab. So here, this is for chain A. I see things, so remember gray it's unmodeled, so it's in the structure, it's just not there in the model, but it's in the actual like protein that they were looking at, but they just couldn't see it, that type of thing. You'll see information about clashes. So in the structure, are there clashes? So basically amino acids that are trying to occupy the same space. You'll see information about how well the model fits the map, and you'll also see then just more general information about amino acid properties, about variants. So sometimes there'll be different places, like this amino acid might be different in some people. If this was, say, a human sequence, maybe people have different amino acids at that place. If you have, if there's changes made to the protein, then you'll see them modified residues. And so you can see in this case, it's telling you that there's a modified residue here, and you could go find more information about it. Regions. So there are various regions that will be, so you can see this is a domain. You can find more information about the domains in that annotations. Okay, and then you have, this was for chain A, so the protein. You can do something similar for the other chains that you have, so in this case, the RNA. Again, there was an unmodeled region, so that's going to be in gray. But then I see information about the quality of the model for the RNA in those other places. I can find where in the genome the gene for this is, and then I can find information about different versions. So that's the basics of what you'll find in a PDB entry and how to navigate through them. Each entry will be a little bit different, and so if you had a cryo-EM entry, that would be different from this. You would have a little bit different of the experimental data parameters, but you'd still have similar things in terms of what you could see with the structure, what you get with the annotations, as well as like genome and sequence and versions. You won't get quite as much stuff if you're on something that is computationally determined, but you do get information about the quality of the model in terms of how confident it is, and you can go and you can download the files for it. And again, use those cautiously. Not that there aren't problems with experimentally determined ones, but there are, at least we know that's like one snapshot, it just might not be all the snapshots. If you're signed in, you can actually save queries, so you can save searches. There's lots of really cool stuff you could do. And if you want to learn more, go to the PDB 101 and they'll teach you a lot more. If you want to get a guide to explain understanding the data, they've got a really great one. So, hope this helps and happy exploring.